So what is DNA sequencing? DNA sequencing is the process we use to determine the linear order of these DNA nucleotides or bases. And A stands for adenine, G stands for guanine, C for cytosine, and T for thymine. And we want to understand the order of these four bases in the DNA molecules that make up the genome. The DNA sequence of the human genome consists of approximately 3.2 billion nucleotides. And to understand its function, it must be properly assembled and annotated. We want to know what the genes in the genome do. We want to know how they are functionally related and how they are expressed. If we were to look at the sequence of bases in a human genome in size 12 fonts, they would occupy 2,000 books, each book 500 pages in length, with 2,700 letters per page. This is an immense amount of information. It doesn't make very interesting reading on first glance. It's good for eye strain, but it's difficult to look at a DNA sequence and immediately understand its function. The reason for that is these four bases, the A, G, the T, and the C, represent the alphabet of the genetic code, but they're written without capitalization, without punctuation, without spacing, without paragraphs. There's no indication in the DNA sequence itself of its function. That is the role of genomics. So how do we determine a DNA sequence? Well, the first thing we do is we take a purified DNA molecule and we fragment it into millions of small pieces. You can visualize that in the slide here as that nest of straw on top of the DNA sequence figure. These millions of small fragments are individually sequenced. They're called sequence reads. Each of them is only a few hundred bases in length. The next step in deriving the genome sequence is to compare the sequence reads to identify regions that overlap among sequence reads. The overlapping reads are then assembled into contigs. It's represented by the dark line here. And then overlapping contigs are assembled into scaffolds, and scaffolds then are assembled into whole chromosomes. This is the world's biggest jigsaw puzzle. If you can imagine a jigsaw puzzle with something like 100 million pieces to be assembled into a single image. This is a video that hopefully will show us how this process occurs. So we're looking at a collection of letters here, and the intent is to identify meaningful terms within this 3 lines of letters. And you can see that they read, "The quick brown fox jumped over the lazy dog. The dog lay quietly dreaming of dinner." This is roughly equivalent to the task that faces scientists as they try to understand the genome.